Welcome back. This is our 15th and final lecture of the semester. We're going to have preliminary project presentations today, and then I hope we will talk about links and compartmental cells a bit. I should say that the links and compartmental cell slides were developed by Giulio Belmonte of North Carolina State University and Priyam Adyapok of Duke University. And I thank them for letting me use them today. As always, this class is live streamed and recorded. And for students, if you want me to edit out your presentations or mute the recording during your presentations, please feel free to ask for that. I think it's useful to be able to see what you presented, but if you don't want it shared, there's certainly no obligation to do that. So we're going to start with student presentations. We'll go over compartmental cells, which we introduced a little bit last time. Alex is a big expert on that, and Pedro also a little bit, more than a little bit, I think. So you may have to correct me if I make a mistake when I present compartmental cells. Then we'll talk about links or focal point plasticity just a bit. We're not going to have time to really go into the details. These are quite powerful techniques, which we cover more in the summer. And then I hope we'll be able to build a few models using those tools to get a sense of some of the other things that CompuCell can do. So to begin with, I'd like to start by having the student presentations. Again, this is a dry run. The goal is that you'll present this week, and then next week during the regular class time, you'll do the final formal presentation. And then also, I'm asking you, since there are only two teams, it's rather easy. I'm asking you to meet the two teams together. Each one of you should review the other team's project. I gave you a sheet which you could use to score the other project. And the purpose of that review is, I said score, but really the purpose of that review is to help the other team improve their project so that that people are able to make changes that would be useful and helpful between this week and next week. I realize that a lot of classes you don't do 2 rounds of presentation, and normally I would actually have asked you to present a week earlier so that you had a little more time to do updates, but it's okay this way. In classes like architecture and in many engineering project courses, you'll have 2 rounds of presentation. You'll do a preliminary presentation, you'll get some feedback from your classmates, and then you'll work on it and do a final presentation based on that. It's difficult to do these big projects in one round and to think about everything that you need to do to present. And so having 2 opportunities to present is something that I think is an important part of learning how to do these kinds of projects. All right. So with that, I will turn over the screen share to William and Satoshi.
Okay. So I prepared the presentation as a poster form because I didn't know that which kind of format you wanted, so I just tried to do it as the other class I'm having. And there's like a QR code in like the bottom right side, which is kind of like a short video of like what the stimulation would look like. And I'll give the time to Shintoshi.
Okay. Our project is we studied the tumor cell in the medium with different microenvironments using the CompuCell 3D. And we have used usual cellular pods model simulation. This is a kind of project that can be used in real life applications for like drug research, for cancer research, and like tissue engineering for regenerative medicine. And it can also be used for like wound healing technologies in the developmental biology, disease modeling, etc. So we can start with the,
Okay.
yeah. Can we see the simulation?
Oh yeah, simulation is over here.
Okay, so this will be our simulation. We have started with the 500 lattice size. And we used a cell type named tumor. Can you open the XML code? Yeah, so we have used boundary conditions as periodic and the temperature as 10. And we have used Monte Carlo steps that are more, okay. We have used contact energies for the mediums as 0, and for the medium tumor, we did 12, and for the tumor cells, we did 8. And we have put our diffusion coefficient as 0.1 and decay coefficient as 0.0001. And we have used blob initializer and with the radius of 100. And can we go to the steppables? So here we used 4 steppables, 2 for the plotting of graphs. And the first one is the constraint initializer steppable. This class will initialize the cells with the target volume and the addition properties. We used our, we've given the cell target volume as 25 and lambda volume as 2.0. And the next step of next class is the growth curve steppable that we used for the graph to plot the total volume growth curve and Monte Carlo steps on the x-axis and the total cell volume on the y-axis. And a histogram plot with the cell volume size on the x-axis and the percentage of cells on the y-axis. The mitosis steppable is the main key for cell division. It would check if the cells meet the criteria to divide, like reaching a certain volume. When the cell divides, this would be called the set properties for the parent and the child cells, such as like doing half the parent cell's target volume and copying certain properties of the child cell. And in the cell motility steppable, this mostly controls the cell movement within the grid. Yeah, its step function could contain like assigning random vectors to each cell, influencing their movement and motility. So this is our code. And can you look at our graphs? So, like in our analysis of the tumor volume over the time, it has revealed a progressive increase in the overall volume, indication of the cell proliferation. But however, the periodic fluctuation suggested phases where the tumor adjusted to the microenvironment constraints, such as decay and maybe some like accumulation. It also says that tumors are adapted to the changing conditions within the environment. Whereas the histogram says that like we saw a broad distribution of cell sizes reflecting the heterogeneous cells varying across the population. As the simulation progressed, the distribution became more skewed, I say, towards the larger sizes. That says that selection pressure favoring the larger cells, possibly due to limited resources, I might say. So yeah, this we are still doing a project, and there are still more insights to look at. This is what we did till now.
Okay. Was there any more you wanted to talk about right now?
That's it for now. Yeah, we're still working on it. So
May I just ask, maybe for a final presentation, it would be helpful if you could explain a little bit more about the biology here. And what are the cells representing? What are the chemical fields representing?
okay, we've used our medium as the extracellular matrix, and we have used a tumor cell. But yeah, we should be studying more about the biology context here, and yeah, we'll be talking about that in the final presentation.
I mean, I understand if you're working on it right at the moment. I'm just suggesting a few things that you might want to consider when you write it up for next week.
Okay, yes, sir.
If you come, can you show the simulation again?
Yeah.
The actual where you in player maybe. Right. So one thing you might want to think about here is, are there parameters in the simulation that a user could change? Maybe the size at which the cells divide, or how much they take up the growth factor or the nutrient, or how often they die, or what the motility is when they become motile. What parameters do you think a user might be able to change, which would help them understand tumor function better? It's interesting to me that I heard a talk this morning by one of Paul Backlund's students, and they have a little simplified cancer simulation where they have simulated cells growing and dividing like this. But when the cells become hypoxic, when they have not enough oxygen, then they become motile and start wandering off. And so I think that Paul's has a version of that simulation running actually on NanoHub. And so you might want to look at that simulation and see how it relates to this one. Maybe you could compare a little bit about the difference between this simulation and the other one. It also might, I think, be helpful if you could talk a little bit about what is it you're trying to learn from this simulation? In other words, you're modeling a tumor growth, which is great, but what is it about tumor growth that's interesting to you? And I think the source paper may have that in it to some extent, but you might want to read a little bit about other cancer models or tumor models and try to ask the question, what is it specifically that you gain by doing a simulation? In other words, what is it you learn from the simulation that you might not know just writing a description of the simulation when you actually run it? Is there something special that happens that's interesting that would be informative? So those are just a few of the things that you might want to think about when you're revising this, and I'm sure Emma and Pedro will have some good suggestions for things that you might want to do to make it better. You've got a good, solid, basic simulation here running, and now you want to think about how to present it in a way that people really learn from it and understand.
Yes, sir. Yeah.
Okay.
Yeah, I'll try to improve it.
Great. Anything else, anybody else in the room? Pedro, Alex, do you have any feedback or Emma you'd like to give now? It could be just anything about that might help them improve their presentation a little bit and make it more useful for potential users down the line.
I think it looks great. The only thing I will do is in the poster, put maybe 1 or 2 phrases explaining the result, and that's all. I think it
You
looks good.
mean in the graphs or?
Yeah, in the side you have a little bit of blank space there. I would explain what the graphs are showing, like in 1 or 2 lines. That's all.
Okay,
Yeah, because some people would like not understand only from looking at it.
mm-hmm.
But I think it's good already.
Okay.
Yeah, thank you.
And I do have just a logistical question, which is, in the original assignment, there was a description of what you were asked to turn in, and of course you may turn in something different from that, but and then I sent out another reminder about what the format of this presentation was, and so I just wanted to make sure that people received that, because if you didn't get that email, and that's I know Hayden got it, but and it was also up on Canvas, part of it. So I just want to make sure that there wasn't a problem with the communication. But
Because like I got the email, but I didn't know where is the template and stuff, so I just like
okay,
yeah.
okay, that's fine. I just wanted to make sure that you received it. That's all. Okay, thank you very much. That's great.
Thank you.
And now, team 2. Let's we'll do a cut slide for a second. So there's team 2, and let's hear from team 2 about what you were working on.
All right, I'll open the presentation. Share the screen. Do you guys see this? All right. So as we split, Emma will talk the first introductory slides, and me and Alex will go over methods and results.
Hi, everyone. Can you hear me all right through this microphone?
You sound fine, thank you.
Okay. In our presentation, we're working on replicating a certain model of the spread of a virus through a culture of respiratory cells. So they're bronchial epithelial cells particularly. Would Pedro mind advancing the slide? All right, so the motivation behind the original paper is specifically to develop a cost-effective way to do a bunch of experiments on influenza and specifically the possible treatment of influenza. So the type of experiment this simulation is aiming to capture is called a viral plaque assay. In this experiment, you take some cells from your host organism that you're studying, and you grow them in the lab into a sheet. So we're in 2D. In this case, the cells are taken from the lung. And once you have your sheet of cells, you take a little bit of the virus you're studying. So here it's influenza, and you expose those cells to the virus, but at a very low concentration, just enough to start the infection process. And then you watch the infection spread through the cells as the cells talk to each other with their immune response and succumb to the virus. So I expect that all of you know that the way viruses reproduce themselves is by getting into a cell and hijacking it, then using the cell to produce copies of themselves, which they then release from the cell. Those copies then go through the extracellular matrix, which is just the space between the cells, to other cells and infect them. And those cells in turn replicate the virus, then release it. So the infection will spread. Of course, once the cells are all dead, the virus can't spread anymore. So there's this trade-off between the progress of the virus and the health of the host tissue. One type of treatment that these types of experiments are being used to research is immunomodulation, having to do with like treating the cell culture with some of these immune signaling molecules. and seeing how much one has to use to potentially curtail the infection. Now, of course, there are potential problems with this method, like you can very much irritate the tissue because some of these signaling molecules can produce very dangerous symptoms. Like, I don't know if y'all have heard about the cytokine storms that people saw during COVID, but I think that that was an important cause of death from COVID. So it's very important to really tease apart how much of these signaling molecules can be applied to improve the immune response and stop the infection before the host is actually harmed. So it's really important to get methods for running these experiments, in silico instead of in vitro. I'm not sure what's going on this side. To improve the experimentation. So one of those modeling approaches was done using CompuCell 3D, and that's what we focused on here.
I have a question. When I open the chat, what happens? Does it appear?
No.
Okay.
So what we did together is we went through this paper and we downloaded the code. We checked to make sure we could run the code, which of course is an important sanity check, since the goal of this paper is to provide a tool that a lot of different researchers can use on their own. We then looked through the code and tried to figure out how we could make it faster, because one of the downsides of this code is that for various reasons, it's quite slow. So Pedro and Alex will tell you all a bit about what we tried to do to make it faster. Then we made it even more user-friendly by adding sliders so that researchers can, without changing the code, because wet lab researchers might not necessarily know about how to use CompuCell 3D or how to program in general, we can now have people go in and play with the simulation by changing the parameters with the sliders. And of course, we wrote up some exercises that can be done using those sliders to help teach people how to use this simulated experiment. And we also tried to reproduce some of the figures from the initial paper, which is another important sanity check on this code.
All right. I'll explain the model structure, more or less how it works, how it's implemented. So starting with the environment, it's a cell lattice, 300 by 300 pixels. Each cell is 3 by 3 pixels, so it's 100 by 100 array of cells. And there are two fields in the simulation, the virus field and the interferon field. They diffuse decay across the environment and can be secreted by cells depending on their state. There are four cell types or cell states: uninfected, eclipse, virus releasing, and dead. The uninfected are cells that can be or not be exposed to viruses, but they don't have any virus inside them. Eclipse cells are cells that have the virus inside them, but the virus is not replicating. And the virus releasing phase is when cells have virus replicating inside them and are able to release viruses into the environment. Dead cells are cells with no virus inside and nothing going on inside, nor secreting, nor absorbing. There are two classes of cellular processes: the intracellular processes, represented by ODEs. They model interferon signaling, virus replication, virus sensing, and cell health, and the cell type transitions. So according to the environment and the intracellular processes, cells may transition between states. For example, from uninfected to eclipse, which is I1, is determined by the local extracellular virus level. From eclipse to infected, virus releasing cells, it's a time delay. And from infected to dead cells, it's mediated by cell health and the level of intracellular viruses. The two scales, the intracellular scale, the cellular scale, and the environment are coupled using CompuCell 3D. So in this figure extracted from the original paper, they show the intracellular pathways. You can see virus replication with the arrow pointing to the node itself, cell health, and virus sensing, interferon signaling. This is coupled to the extracellular environment through CompuCell secretion and interfacing the intracellular parameters with Python, the Pythonic side of CompuCell. So the level of interferon, extracellular interferon, is an input to the intracellular model, and interferon is a field that diffuses through the space. We try to improve the simulation at first by not changing the model itself, because whenever you change a model, you run through some risks of changing the outcomes. So I limited myself to reducing the number of for loops. So previously, for every mechanism and process taking place, there was a for loop. So there was a for loop for all the cells, then another for loop for each one of the types, and pairs of types combined. So my modification was putting everything in the same loop and if statements to acquire each cell type. And also we deactivated the file outputs for the sake of learning with the simulation, which is the final purpose of the project. We don't need file outputs. We don't need huge text files with cell positions, etc. So we deactivated that. And so we get some performance to be able to play the simulation for learning purposes. We added sliders and plots. We tried adding more plots, but the plots are expensive in terms of computational performance. So we chose 3 graphs, 3 plots: the average extracellular interferon level, the average extracellular virus, and the fraction of cell types. So how many of the cells are uninfected, eclipse, infected, and dead. And we made a selection of parameters that the user can change. And the user can change by multiplication. So if the value is 1, it's the original parameter. You can select 1, 0.1, 10, 100. So you're not setting a parameter because most of the time the user doesn't understand what does it mean a diffusion of 0.1 or 0.2. So instead of setting the exact value, I opted for setting a multiplication factor. And this idea is not mine. This idea is from the paper itself. In the paper, in some graphs, in some figures, the authors multiply the parameter by 1, 10, and 100 and compare the different scales. So we selected interferon diffusion, virus diffusion, interferon and virus decay, and some key parameters analyzed in the original paper, which are the rate of health loss by virus, stat activation, and RIG-I activation. And finally, Alex will take over. We selected 2 results to try and reproduce. One for high MOI and another one for low MOI. So I'll let Alex explain? Go ahead, Alex.
So I say one thing to add. So MOI stands for multiplicity of infection, and that's the ratio of virus particles to susceptible cells. So at a really high MOI, that means there's a lot of virus for every cell, and that those cells are probably not going to survive in general. So for the first part, we just demonstrated that at a high MOI, all of the cells are going to die, and that even though that there is a lot of interferon being excreted, it's just too late for the cells. They're doomed. Over the course of a few hours, all the cells will go from alive to dead pretty rapidly. So then the graphs just showing that over time, the number of cells are going to die. The virus will go down as those cells die. Even though the interferon will spike, it's too late. And, yeah. But and also like the intracellular IFN, IRF7, STAT-P, all of these cellular responses to virus are just not enough. Even though, so like the cellular pathways that would try to prevent viral replication are there, it's not enough. Die. And then to the second part, that if these cells are not stimulated with the interferon representing the antiviral cytokine, the virus pretty much spread uncontrollably and all the cells will be infected and they will die. The virus will increase, and although interferon will increase, it's not enough. Like interferon will naturally increase as the cells are infected and die. Or most of them die. And then if the cells are stimulated with interferon, the virus will initially increase, but it'll kind of plateau and then immediately and then plummet as the antiviral response kicks in and prevents viral replication from occurring. This is the link to the code on GitHub. And the citation.
So yeah, this is our summary of our project. In the GitHub, we have the exercises that guide the user through the simulation, understanding the parameters. And I can run the simulation, but I think it's all explained in the slides, so I'll end the presentation here.
Well, if you've got it on your computer, maybe you should just, if you're willing to, just show it running. That would be.
Okay.
Do you think it will crash your computer or not? With
I
Zoom
think
at the same
it
time?
will not crash my computer.
Mine with Zoom running, it takes up so much of the CPU.
Yeah, when I try to run mine with Zoom, it just becomes really slow. It'll run, it doesn't crash, but it becomes incredibly slow.
I think I opened the wrong one. Let me see.
Oh yeah, you might want to, if you're modifying the model, you might want to update the version of Compusol it's expecting at least, so it doesn't throw a warning on that. So were you able to get an estimate for how much speed up you got from the changes you made to the code?
Yes. And I was confused. I thought I was running the wrong version, but actually it takes a while for the graphs to show up. So this is what, yeah. Yes, I did some estimates, and basically the changes I made were useless in terms of performance. And to really speed it up, I have to either the only way to speed it up is by removing one steppable class entirely. The steppable class that contains both the loops and the intracellular model. If I just delete the intracellular model, it's not enough. It will speed up by a little bit, like it will be like 50% faster or something. If I deactivate everything, then it becomes like as fast as a normal computational simulation.
I would have thought the thing that was slow here was the diffusion.
I don't think so. When I deactivate the steppable class, it runs okay. It runs like 10, 20, 30, 40, 50, like really quickly.
That's interesting. I wonder what's happening. That might be one to take to Maciek, because normally calling steppables by itself doesn't take that much time.
Yeah, so here we can, for example, let's increase virus decay and see what happens to the extracellular virus. It's high. If I set to 100, we should see it going down eventually. The simulation has the spread has phases. This is really interesting. So eventually the cells will secrete a lot of viruses, and then sometimes not as much. But this is 10 to the 6. With virus decay of 100, I was expecting it to decrease, but maybe it's too late already. I think it will decrease, yeah. But it could be too late. It depends on when you do it. If you do that in the beginning of the simulation, it's very obvious. But I think this will work eventually. The viruses will decay.
Do you have a glitch where the cell field is not updating? No, there it is. You have to click on it to get the cell field to update.
I don't think so. It updates every 10 Monte Carlo steps, so yeah.
But maybe you show the viral field and the interferon field just to get a sense of what those look like.
Yeah. So interferon field is mainly secreted at the border of the cluster. I mean the not a cluster, but this is like a wave, let's say a wave of interferon, because the cells inside the infection circle, they are dead, they don't secrete interferons, and the cells at the border are exposed to viruses and they can secrete interferon. So we see a wave of interferon expanding, and the same thing for viruses. The cells in the middle die, they cannot get infected again, so the virus field decays quickly. And this wave spreads much faster than the virus can diffuse. So by regulating diffusion and decay of interferon and virus field, you will play with the race between interferon and virus, and you can make interferon reach neighboring cells much quicker and effectively than viruses, and study how they race through space.
Okay, great. Any comments from Santoshi or Hayden or William? I mean, this may be one case where using that trick of having a pull down menu in the steering panel that starts and pauses the simulation might be useful. So in this case, the simulation will always start with the default parameters, and then the user can change it after the fact. But you might, for example, be able to change the MOI. And that would be something you'd have to do at the very beginning. And so you might want to have a controller that pauses the simulation at the beginning until the user pulls down something, say start simulation, for example. As a little bit of a, again, it's mainly doesn't change the science of the simulation, but it changes the user interaction a little bit. But it doesn't seem that slow. I mean, you're seeing the plaque grow in a time scale where it's fast enough that you don't go crazy, and it's slow enough that you could actually see it. If it ran too fast, you wouldn't be able to see what was happening. So, okay, great. Well, I'll be very interested to see how this develops between now and next week then. And I think this could be a nice simulation to put up on NanoHub. We're not doing NanoHub this year per se, but it's the kind of thing that could be pretty informative, I think, as a NanoHub app. Okay,
Is there
anything?
any other way to speed it up, Pedro?
Sorry?
You have any hypothesis about how to speed it up?
No.
Well, if you really believe it's the steppables that are causing it to slow down, you could set it so the steppables are called every 10 Monte Carlo steps, for example, instead of every Monte Carlo step. So you could do that. That might be possible. Depending on how fast the viral replication is and how fast the infection is, you might do that. Again, my recollection was with this simulation, the problem was that the diffusion constants of the virus and the interferon were quite fast. And so the diffusion solvers are slow. But I'm interested that it's you think it's the steppables that are slowing you down. You could always do it. Maciek taught us how to do it and rewrite the steppables in Python. In C, from Python to C++. That would speed things up. Of course, it makes the code less shareable. That's a possibility. Okay. Well, thank you. Any other comments? Sorry, Hayden, go ahead.
Yeah. Can I see your first slide again?
Yes.
Well, so your intro slide, obviously. I didn't have too many comments about your title slide. Let's see. Yeah, so I think that this kind of what Professor Glazier was saying to William and Santoshi as well. One comment I have, so this is a good setup of why you're doing what you're doing, but I think what both groups could use was just a couple of slides explaining the actual physical system that you're looking at and the biological background. Because you kind of jump straight from this and the next slide into the model. But for people who aren't familiar with kind of exactly what you're trying to model, having a couple of slides just saying, this is what we're looking at. If there's a couple of pictures maybe from experiment to give it some context. Particularly once you start explaining the network model stuff, having a little bit of that biological context will help make some of this make a little more sense to the uninformed. So I think that was my main comment, but otherwise I think it looks pretty good. Yeah, sorry, I said like I was going to say something else, but I didn't actually have an end to that that sentence. But that was my main comment for both groups, is that just a little bit of explanation of the biological system, kind of more from a top level, would be helpful. Kind of bridging that gap with how the network model relates to the system itself, if that makes sense.
Yeah, I agree. Yeah, and I neglected that. I think I was in a rush and I didn't realized that it was lacking the biology background. I'll add that.
Mhm. But yeah, otherwise I think that that's looking good. So
Okay, thanks.
Okay, thank you both teams. And I know this end of the semester is always hectic, and so try to get these things pulled together. But you've got a lot of projects is work, but I think having gone through it now, maybe you'll have ideas about ways to improve for next week. Take the code you've written and think about how to present it as effectively as possible. These are some exercises exploring compartmental cells and explicit force links that were developed by Giulio Belmonte and Priyam Adyapak for our summer program. And these are teaching a little bit more sophisticated spatial modeling than we've done so far in the class. And it's building on what we were doing just at the end of the class last time. So examples of this kind of simulation would be the things that we're seeing here, where we have compartmental cells. In this case, we have what's called epithelialization. Not sure why I'm tripping on that word. I can normally say it just fine. In which the cells polarize. And this is what happens when cells form sheets, layers. We think of ourselves as three-dimensional, but in fact, we're really composed of a lot of two-dimensional layers that are organized in a variety of ways. They can be rolled up to make tubes and branch structures. They can be layered on top of each other. But much of our body and many of the cells in our body are organized in what are called epithelia, that is sheets, with a relatively limited amount of what's called connective tissue, which is really three-dimensional tissue in between. Often you'll have what are called complex epithelia, where you'll have one layer of cells on top of another, something more like perhaps corrugated cardboard or plywood, not single layers of cells. But there are plenty of tissues in which you do have a single layer of cells, the lining of the lung being an example of that, the lining of the blood vessels being another. In all of these, we have to distinguish what's happening at the top of the cells, what's called the apical surface of the cell. Very often that's not adhesive, or it's exposed, so it'll be exposed to blood, to air, to other fluids. A basal layer, which often binds to what's called a basement membrane, which is an extracellular fiber structure, a little bit like a woven mat that the cells pull on and keep under tension. And then what's called a lateral surface or a mediolateral surface, where the cells adhere to their neighbors. And so there are really three functional regions in an epithelial cell, sometimes more than three, but at least three. A basal surface, which binds to the substrate with special adhesion molecules, normally integrins. An apical surface that often secretes or absorbs from the outside. and then the lateral surface that's responsible for the mechanical integrity of the tissue. Here, in the case of ectopic somites, we start out with cells that are non-polarized. Those cells polarize. They send a signal to their neighbors that say to co-align. If the cells are misaligned, they depolarize and repolarize. And because there is a contraction on the apical surface, a little bit counterintuitively, in this particular case on the bottom left, the red is actually the basal surface that represents the basement membrane, and the green is the apical surface. Very often there's also a tension in the apical region of the cell that's organized by actomyosin, motile molecules in the cell that have a contractile function. And then in this retinal neovascularization, we have the pigmented retinal epithelium in green, and then the multi-layered structure, which represents the photoreceptors on top of it. As we saw last time, cells have a lot of different shapes. They can be polarized in a direction where they're crawling. This would be typical of something like a fibroblast or a keratocyte. And epithelial cells have, we saw the basic architecture here. This is digging a little bit more to what's happening biologically in epithelial cells. In this case, there's cells that are specialized for absorbing from an extracellular fluid. There are tight junctions that bind the cells to their neighbors. There are also desmosomes that bind cells to their neighbors, and a variety of channels that allow molecules to flow back and forth between neighboring cells. And we could also use this kind of architecture to represent actual physical structures of the cell. For example, we could represent the nucleus as a separate domain within the cell. And as I mentioned, partly because of the history of CompuCell, we don't change anything in terms of cell ID when we're going to build compartmental cells. We take multiple cells with different ID and assign them what's called a cluster ID, which is really the ID of the cell containing them. And so when we want to build compartmental cells, every cell has now two IDs. Its individual numerical ID, which is unique for the whole simulation, just as before, and then a second ID which says I'm part of cell number 55, cluster cell number 55, and that's called a cluster ID. When we do that, something that is a little bit awkward sometimes is that a lot of the CompuCell functions don't know about clusters. For example, cell type doesn't know about clusters. And so sometimes we're going to have to iterate over clusters and look at their components by hand to be able to make changes. But effectively, if we want to cluster different compartments together, we have to assign the same cluster ID to all the compartments we want to be part of the same cell. You could do rather elaborate things with clusters. There's this beautiful simulation of myxobacteria from Deutsch's group, where they have a chain of basically beads on a string. They apply a backbone through that chain, and then they assign a bending energy to that backbone. And that does a pretty good simulation of myxobacteria or any other rod-shaped bacteria. And then there's this nice simulation of a boss doing eye cup formation or lens formation with invagination. The simulation I hope we'll get to today is the first step in trying to build a simulation of the kind that we have here. We're not going to get all the way through that in 2 hours, but you'll have all the pieces, and if you wanted to go forward and replicate this particular simulation that you're seeing A, B, C, D here on the right, you could do that. The key thing that is going to be different when we're using clusters is that the contact energies are going to be different for two elements of a cluster, depending on whether you belong to the same cluster or different clusters. So, for example, the nucleus in a cell has low energy with the cytoplasm surrounding it, but it ought to have very high energy with the cytoplasm of another cell because it should never come in contact with that. And so I'm going to have the regular contact energy, which defines the contact energy between any two components that are in different clusters. That's what we're used to. And then a separate contact internal, which says what the contact energies are for those components within the same cell cluster. And that's the thing that we sometimes have to get used to. You may very well find negative values. In principle, you could have negative values in regular contact energy too, although we haven't used those very much in this class. But you may very often find negative contact internals within this system. The next thing that I want to talk about, that's what we covered a little bit at the end of last class. We didn't really do a big exercise on it. The next thing I want to talk about is something that is useful even without cell clusters, but becomes especially necessary when we do have cell clusters. And I apologize for, wonder what happened to the formatting on that slide. It's interesting because I don't have that format error in the when I look at it. Here, let me fix that so you can see. Is the screen share still okay?
Yes.
Which are what are called links or focal point plasticity links. And these are essentially springs, although you can give them properties that are not spring-like if you prefer, which connect the centers of mass of regions. And if anybody has worked with a center model method like Paul Macklin's Physicell or T.J. Sego's Tissue Forge or Biocellian or Episim, these are all other simulation frameworks, you define very often particles, as you would in a molecular dynamics simulation, and then links between the particles, as well as potentials. So those are what are called bonded interactions. Say, if you have two atoms that are in a molecule, there'll be a special link that will describe the interaction between them, which would be different from the interaction between the same atom in different molecules. So in a sense, if you've done molecular dynamics, this sort of clustering that we're doing here is very similar to what you do in molecular dynamics when you define molecular species. So in this case, we're going to say that there is a link defined between this cell and this cell here. And probably are people seeing my pointer or not? Somebody said the other day that they weren't seeing the pointer, so let me make sure that that's working.
I don't see it.
You're not seeing it. Okay. Well, sometimes it works in all right, how about now?
Oh, wait, yeah, okay.
Okay, fine. We'll use that. Okay. So we have a cell 1 here and cell 2 here. We've defined a link between them. And that link is going to have an extra energy, which is the elasticity of the link or the spring constant of the link times the link length minus the target length squared. And if anybody has ever done a spring, the Hookean spring, this is exactly the equation for a spring. This means that as we stretch this link or compress it compared to its target, there'll be a restoring force either pulling back or extending. And that restoring force will go linearly as the distance, the extension of the spring differs. So the more we stretch it, the stronger the restoring force. And that's actually not typically how things work in biology. Usually biologically, those restoring forces tend to be independent of the length of the target for large distances. For short distances, there is some elasticity of that kind. And so what this means is this is going to add into all the regular energies, which means that if this cell tries to move to the right and it starts stretching the spring, there's going to be a force pulling back on it, restoring it to its original position. And so we are going to have two parameters associated with each link: the strength of the link, the spring constant, and the target length. Now, we're going to be allowed to define other functional forms for links, in particular one that's independent of length, which just pulls all the time with constant force. And you could build other forms as well. But that's the one that we're going to run with most of the time. Okay. So if we're going to use links, they exist in Wizard. They exist in Twit code snippets. So if you go in that Cell Properties and Behaviors panel, there's a button here, Focal Point Plasticity, which we click. And you usually will need to know who your neighbors are, so clicking Neighbor Tracker is a good idea. And that's going to paste in some XML code. And as usual, sometimes we'll use the XML code, and sometimes we'll do things in Python. But you'll get a plugin named Focal Point Plasticity. And by default, Focal Point Plasticity acts on all cells of a given type. And so you will say parameters and then type 1, type 2. So you could say, if there's cell type A and B, you could define links between types A and A, or type A and B, or type B and B, which are different from each other. Then you have that lambda, which is the elasticity of the spring constant of the spring. Target distance, which is the equilibrium length at which there's no force. The maximum distance, in this case, there's a predefined breaking length. So if you pull on the spring enough and you get it stretched too much, it breaks. And so maximum distance says that the spring breaks if it's stretched too much. I tend not to use that one very much, but that's the default in CompuCell. The other thing that we could do is we could say that for any given cell, there is a maximum number of links that cell is allowed to make. Can't make life, that's an integer, and it has to be at least 1. We tell the cell not to make any links, then it won't. But typically it could be a number like 2 or often 6 because of the number of neighbors of the cell. Neighbor order is there because when we're calculating who's next to us, that depends on the range over which we do the calculation. And the last number, which is a little bit unexpected perhaps, is activation energy. And this plugin is maybe a little bit too smart for its own good. If you have a configuration of cells and you turn on focal point plasticity, CompuCell will try to generate links between the cells in a quasi-random way. And so if you say, I want to have 6 junctions here, it will try to create 6 links with the neighboring cells. And the activation energy basically controls the probability of generating a link with each neighbor. All right. So this is just reviewing the maximum number of links between cells. Okay, and that says activation energy lowers the energy barrier for creating new links, and the neighbor order determines how far you need to be to create new cells. Personally, I tend to do link creation and destruction by hand because I find the automated one confusing. On the other hand, if you're going to just learn how links work in general, it's not so bad. So I would like everybody to fire up CompuCell, start TwitIt, create a new simulation, and we'll walk through this together. If you get stuck, all of these simulations are loaded in the student materials folder. But as usual, it'll be student materials folder, demo, lecture 15. And then there are a couple of subfolders that you might have to look into to find these. The subfolders, I think, are going to be numbered 6.2. And so if you're looking for demo 6.2.1, it would be, you'd look in the folder student materials, demos, lecture 15, 6.2, and then you'll find the subfolders. So let me know when you've got TwitIt running. Right. Okay. So we're going to do a new CC3D project. You could call it, you could see this is coming from last summer's workshop, but give the simulation a name that you like. As usual, no spaces, dots, slashes. Don't start with a number, the usual things. And remember to write it into a directory where you know how to write it. Give it a name. And to begin with, we're going to define, I think actually everything here is default. We'll use uniform initializer, rectangular slab, 256 by 256 lattice, no flux boundary conditions, square lattice, fluctuation amplitude of 10, pixel copy range of 1, 100,000 Monte Carlo steps. Okay. We're only going to use one cell type here. So create a single cell type. We're not going to use any chemical fields. And now in the cell properties and behaviors, we'll need contact energy. We always pretty much need contact energy. Turn on focal point plasticity, volume flex. If we forget to turn on volume flex, our cells will disappear on us. Center of mass, and not a bad idea to check cell neighbors too, which is not marked here. Okay. And now try hitting done and run it and tell me what you get. Did it run or crash? It's the first question. Anybody? Emma, what happened?
I'm still waiting for a player to start. Oh, here we go. Oh, wait a minute. It's running. Yep.
Okay. Okay, great. So you should see something that looks like this. Now, if you don't see something that looks like that, pull down visualization, and there are all these tick boxes which we don't usually play with very much. We've used those a little bit when we did 3D simulations, and we needed to adjust the visualization. But here you need to click on the FPP links option. And if you go to settings here, when you go under cell colors and cell colors, there's cell border color control. That's if you have a light colored cell and the links are shown in white, it can be hard to see. So you may want to control that. So why doesn't everybody take a minute to get that working and make sure that you see something like this where you could actually see the links? And remember that when you do that, you're going to have to hit play again when you change the settings so that the simulation continues. Did everybody get something that looks like that? Alex?
Yes.
You're the link expert, so. How about William and Santosh? Good. All right. So, why do we see what we see? We see, basically, if we zoom in here, that most cells have one link, and occasionally there'll be a cell with no links at all. And we don't see any cells with 2 links or more. And that's because now, if we switch to Twit and open up the XML, we'll see that the default settings were lambda of 10, activation energy of -50, target distance of 7, maximum distance of 20, maximum number of junctions 1. So we're only allowed to have 1 link per cell. So what I'd like you to do now is edit maximum number of junctions to 2, run it again, and tell me what you get. In particular, what can be sort of interesting in this case is what kind of connectivity do you need to make sure that all the cells are connected, or that all cells are connected to at least 3 neighbors, for example. So if you change that number to 2, what happens? Anybody? Did it do anything? Can you see the difference? Okay. All right. Try 3. Right, that looks nice. Emma gave that's nice. You could definitely drop a picture of your simulation into the chat. That's a good way to communicate this to. Once you've done 1, 2, and 3, you might try 10. Now there we go. Alex has a nice one. So you could ask the question, at what point is that network of links connected or not? Is 2 enough to do that? Is 3 enough to do it? Is 4 enough to do it? And you'll see that the geometry will change rather radically as you change that number. If you play with the max distance and that activation energy, the pattern will change, but in ways that are not, those are not so obvious sometimes what happens. If you change the activation energy just a little bit, you're not going to see a big change. If you change the maximum distance just a little bit, it won't change. If you made the maximum distance 3, you probably won't get any links. If you make the maximum distance 100, and the neighbor order is 2, 3, or 4, you won't see anything. You try to make it a neighbor order 15, and you make the maximum distance 100, you'll see that basically all cells will connect with all other cells. Somebody could try that. Anybody have anything interesting they want to share besides Emma's and Alex? So here's some examples. If I actually have a 2D simulation and the cells are all the same size, then geometry tells us that the average number of neighbors is going to be 6. That's true for any range of sizes. But if the cells are all approximately the same size, essentially every cell will have 6 neighbors except for the ones on the edge. If there were a lot of size variation, you might get five-fold sizes, 5-sided cells, 4-sided cells, 8-sided cells, and so on. In this kind of simulation, you might occasionally get a 5 or a 7, but you're not going to get much more than that. So here we see the nearest neighbor, 1 link per cell. It's a disconnected graph. 2 links per cell. We're beginning to get these chains of connected links, but there's still some isolated link chains. And we'll see that occasionally there'll be a cell which doesn't have any links. When we go to 3, that backbone of links is connected. With 3, if you had a very big simulation, you might occasionally get a defect where there's an unconnected cell or an unconnected link, but it's going to be quite rare. When we go to 4, we're beginning to fill in essentially a tiling of the cells. And when we get to 6 or 5 or 7, we're going to see that basically every cell is connected to its neighbors. And so if we were going to do a triangulation to do finite element modeling, this is how we would do it. And in fact, you could do solids modeling in 2 or 3D with CompuCell using links. It's computationally rather slow because for solids modeling, what you really want is just the vertex, and the vertex should move under the forces. Here, you're dragging the cell along with it, so you have to actually move the cell using the Potts algorithm to relax the simulation, but it works. So it's not the best way to do solids modeling, but it's possible. And it's actually rather interesting. If you take this example, say, with 6 links, and you apply explicit forces on the left and the right and pull on it, you could do a stress-strain diagram. Some years I'll do that as a little exercise. And you can explore the Young's modulus of the material, the Poisson coefficient of the material, and so on. You could design materials that have rather interesting mechanical properties. In this case, we're not applying any forces, so you're not going to see links breaking. But if we pull on the material, we could have the links break and reform as well. Okay. Now, pick one of these connectivities. One, I'd suggest maybe trying 2 of these examples. You could have a connectivity of 1 or 2, and then also try 1 each. And then why don't you change the target distance? Make the target distance, the default target distance was 7. Now make it smaller. Make the target distance 2 or 1. What happens? And the best way to do this is not by stopping the simulation and starting it again, but by doing it in the model editor window and player. First, check that that works. In other words, run the simulation, go into the little model editor window on the left-hand side of player, open the focal point plasticity plugin, and change the number of links and see, does it actually change the display? If it doesn't, then you'll have to stop the simulation and restart it. If it does, then you can play with the target length. Try making the target length short. You'll be pulling on the cells, applying attention, and then also try making it longer. Try making it 7, 8, 9, 10. What happens? If somebody has that working and wants to show it, that would be great, because this is one of these things where it's much more convincing to see it in action than it is to talk about it. Alex, how about you? Do you have it?
Yeah, I can show it. Right now it's at 7. Go to like 2. Shrinks. I thought it was a little more interesting when we made it really large.
I mean, it's interesting in this case, if you make the target length long enough and the elasticity is strong enough, you could actually push the cells apart from each other. They'll disconnect. I wonder if you could get that to work. Yeah, here you see it's pushing the cells out of the cluster. Emma is getting the same thing. You could get some pretty wild geometries here if you play with this. And sometimes you could get some things that don't make a lot of sense because you're anchored on the center of mass of the cell, but nothing says that the cell has to remain connected. And so you could tear the cell into shreds, and then the anchor point could actually be outside of the cell. If I had the cell break in two, then the center of mass of the cell would be not inside the cell, but would be between the two components. And so that can happen as well. So you do have to keep an eye out for non-physical behavior here. That's great. All right. So here's some examples of that. We just saw Alex do exactly this. When we make the target length very large, we push the cells apart from each other. We can get links that cross each other. The links are not volume excluding. The links only break if they exceed the maximum distance that we set. Now, if we were simulating something like making these links represent axons, it wouldn't be crazy, in fact, to have the axonal pathfinding region, the growth cone region of the axon, be essentially physically separated from the soma of the cell and have a narrow link that connects the two. So it's actually not unreasonable to have that kind of picture. But of course, we want to know why we're getting the picture we have. We don't want to just have this happen by accident. Okay. So that's what we could do with the basic link plugin by itself. As usual, to do something fancier, we'll have to use Python. And the good news is that unlike the volume constraint in CompuCell, where you either could have the volume constraint specified in the XML or in the Python, but not both. Since this was written more recently, it has a tag called local, which says use the XML values to initialize the simulation, but you are allowed to overwrite those values in Python. And so that gives you a little bit more flexibility. One thing that you probably want to do if you're going to do Python specified links is that you actually want to set the maximum number of links in the focal point plasticity plugin to 0, so that the automated link generator isn't trying to generate links for you while you're trying to do it by hand. It can be pretty confusing when you have both of those things running together. And so you want to add that local tag to your code. Okay. If you look at Twit, as usual, there is a menu, a code snippet menu under focal point plasticity. And also, as usual, there are probably too many options. And so finding the thing you want is probably the most difficult thing. There are a lot of things that one wants to do with links, and because they were set up late, the things that are set up automatically for links are not as developed as they might be for some of the longer established plugins. But if we look under the links, we'll see a variety of things called link inventory. You might want to know, is there a link between two cells? That's a useful thing to know. You might want to know, what are all the links that connect to one cell? The endpoints of the links are called anchors, and CompuCell allows you to have fixed anchors that are not inside of a cell. You could basically tack a link down to a point in space that's not inside of a cell. The one thing that you can't do, which would be useful, would be to have that anchor point move. The moment you've done that, you've written a full center model simulation code. And in fact, the whole package of TissueForge started out as an extension to CompuCell to allow links and anchor points to move. So that's how that happened. We also, as we were talking about, links are particularly powerful when they're used in conjunction with compartmental cells. And so it matters a lot whether a link is coupling components within a single cell, for example, the nucleus to the apical region of the cell, versus a link between, say, two apical regions of neighboring cells. And so you'll see that there are options for get number of links for internal links as well. We could create anchors. Those are external points which we will have one end on the anchor and one end on a cell. That's tethering a cell to a specific point in space. We can create FPP internal links, which only work within a cell in a cell cluster. We could create FPP links. We could delete links. We could delete all the links attached to a given cell, if you like. And then we could change all of those properties that we were just looking at in the XML on a cell-by-cell basis. Another thing that we would want to do is we need to be able to iterate. We need to be, say, able to do something to all anchors attached to a given cell, to all links attached to a cell, to all cells attached to a link. And so we have to look through it. I recommend pulling that menu down and exploring one at a time what that menu can do. So if we want to create an FPP link, if we want to create an FPP link here, link management, create FPP link, that will paste in the code link equals self.new FPP link. And then we have to tell it which cells we're connecting. Cell A, cell B, and then the target distance here and the lambda. And that will create a new link. And if I'm doing the simulations, that's how I do it. I always do these by hand. Another one that's pretty useful is remove all cell FPP links on a cell. That'll break all of the links attached to that cell. Okay. So the next model that we're going to work on is a little model of cell crawling. This is the cell crawling in a tissue. A lot of cell crawling models are representing cells moving on a substrate without other cells involved. This is actually how cells move within tissues, within dense tissues. And a typical example of this would be neural crest cell migration or lateral line migration in fish. Again, on a flat surface, cells move in a particular way by extruding lamellipodia and then detaching. Inside of dense tissues, cells move rather differently. And here you see cells crawling as a cluster. This must be lateral line. It's not labeled, but this is actually the formation of the lateral line in the fish. The lateral line is evolutionarily related to the hair cells in your inner ear, except in fish, the lateral line is displayed along the side of the fish in the water. And it's what fish use to detect movement of prey and predators in the extracellular the environment around them. And these are going to be sensory cells that then connect to the nervous system of the fish. So Emma, this is actually a neural development, a real neural development movie. The details of how those clusters of cells are left behind as this cluster moves are still not terribly well understood. There's a group, for example, at National Institutes of Health that's spending a lot of time and effort trying to understand that both experimentally and computationally. This is a picture of cell migration inside of a tissue. Again, if you were to compare this to the way cells crawl on a substrate, when they're by themselves, the way the cells move are rather different. You'll see cells are rather elongated. You also see that the cells will branch and they'll explore multiple options. You'll see the sort of what are called growth codes, where the cell will extend protrusions in multiple directions and then decide on one direction and move in that direction. It's a bit hard to see at this level of resolution, but you will see sometimes little, very thin protrusions from the cells. And here's a little example of that. This is a picture of a single cell, and you'll see these fine, almost lightning-like protrusions. Those protrusions are full of actomyosin. So there actually are fibers, and then eventually also microtubules. There are fibers that are extending what are called filopodia, philo like thread, podium like foot. So these are thread feet, and these are extended and retracted quite rapidly by cells. If you're able to watch them under sufficient magnification, the cells are continually sending out fibers and pulling them back. These are almost always under tension. In other words, if you want to think about how they work, think about having a rope with a grappling hook on the end. The cell's continually throwing out the grappling hook and then pulling back. If the grappling hook doesn't attach to anything, it'll just pull back, and the grappling hook and the rope will come back. On the other hand, if the grappling hook attaches to something and I keep pulling on it, then I'll get pulled forward to wherever I grappled to. And so that's really how these cells move. They throw out these grappling hooks in the form of filopodia. At the end of the filopodium, there are molecules that recognize their target, whether it's another cell or extracellular matrix. If those molecules attach and anchor, then the contraction will actually pull the cell towards the target. And some bacteria move this way too, depending on there are a variety of different bacterial movements. And so here we're going to have a single cell. It's going to throw out filopodia. If those filopodia are able to anchor onto one of the neighboring cells, then it'll pull, and that'll pull the cell forward. Okay, that's what we're going to try to do. When cells are polarized, there is what's called an opening angle. Typically here you'll see that there's some angle. This is the back end of the cell, the trailing edge of the cell. This is the leading edge of the cell. The direction from the trailing edge to the leading edge is the polarization direction. And then the filopodia will get sent out preferentially towards the leading edge direction. Not only in the front, but over some range, typically about 120 degrees. Okay, and so now what we're going to do is we're going to create a tissue, and then we're going to create a second cell type, which is going to be a crawling cell, and we're going to use the filopodia to allow that cell to crawl through the tissue. Is that clear? What we're going to try to do. Okay. So let's have people do this. This is the same set of steps that we just went through. So I'm not going to walk you through all of the individual steps, but I'd like everybody to walk through it. We'll make the dimensions 84 by 35. We'll make them periodic in this case. We didn't do that before. We'll have two cell types. We could call one red and one crawler. No chemical field, contact energy, volume flux, focal point plasticity, neighbor tracker. And that's what you have to do in Twinit, Wizard, hit run, and tell me what you get. William, maybe this time you could tell us what you get. And if you've got that working and you're waiting for other people, you'll see that what you get doesn't look very much like what we want. And so you could begin to think about how to change the simulation to make it more like what we want. William, what did you get? How about Santoshi? Anybody else? People stuck? Emma, how about you? Alex? You're nodding your head.
I
You
have it
want,
working.
you have it working? Okay. You want to show us what you got?
Sure.
Yeah. So you have the two cell types, a random mixture and the links are just sitting there, not doing anything very exciting. If you run it, we certainly don't see one cell moving in the tissue of the other type, do we? So now the question is, what do we have to do to make that into a cell migration simulation? It's not trivial, but it's also not terrible to do. So the first thing that we want to do is in uniform initializer, we really only want one yellow cell. We only want one crawler cell. And so why don't we make all of the cells red? So in the uniform initializer, where you have the two cell types, red and crawler, get rid of the crawler cell. And then in the focal point plasticity plugin, uncomment that local tag and make the maximum number of junctions 0 and set all the contact energies to 2 and run it again. See what you get. One thing that you'll get is there won't be any more links. So you shouldn't have any links. And it should be all red cells. So the simulation is going to be rather uninteresting. But let's make sure it runs. People can give me a thumbs up or a head nod when it works. And as usual, if you're stuck or you need help, we're here. And I apologize, Emma. Somehow I had the chat hidden, so when you said you could share your screen, I didn't see it.
That's okay.
Thank you. Okay, so Emma has that boring simulation now. So let's see what we could do to make this a little more interesting. Okay, so we get this. Not so exciting. So now we want to create one yellow cell. And there are a variety of ways we could do that. The way Julio suggested to do it was to actually loop through the cells inside of the start function, ask where the center mass of the cell is, and if the cell center mass was between 7 and 21 in the x direction and 14 and 28 in the y direction, change the cell type to be crawler. Since it's periodic, we don't really care which cell it is. So another way to do it would be to loop over the cells and take the first cell that we find and make that cell of type crawler. We could go in and find by ID. We could find cell ID number, the cell with ID 3, and make that a crawler cell. That would work too. So in any case, we want to stop our loop, if we're going to loop, after we find one cell, because we only want one crawler cell. So everybody find a way. You have lots of ways of doing it. To change one cell out of all of our cells to be crawler. And let me know when you've got that working. Okay, Emma's got it. Alex?
I'm working on it.
Okay. if you've got that running, you could begin to think about, we'll go over this step by step, but if you want to get a little bit ahead, you could think about how could you create links in a particular direction to pull on the cell. Here's some code that Julio used. So he went over the cell list and then asked the question if XCOM was between 7 and 21, YCOM was between 14 and 28. If so, he changed the cell type. And the break means that once you've done it once, you exit the loop. So if you don't have your own way of doing it, you could try that code. I'll give people a minute to type that. That's a somewhat inelegant way of doing it, but it works. Does anybody need help with that, or is it working for people? So now we have to think a little bit more carefully. We are going to have to define a direction in which the cell is going to move, the angle over which we could create links, the link strength, the target distance, and the maximum distance. And there are a number of different ways that you could create parameters in CompuCell. If you declare things before the class definitions, those values are available everywhere in the simulation. But by default, they're not writable, they're only readable. And so we are going to create global variables, the global parameters, maximum angle, lambda, target distance, maximum distance. And then we are going to declare these as globals. And that global declaration is going to allow us to be able to write and change those values inside of the class. If we don't make them globals, we can read them inside of the class, but not write them. And so I'd like everybody to basically type this code. If you want to abbreviate the variable names, that's the parameter names, that's fine. But we're going to need something that defines that maximum opening angle for the simulation, the maximum length of our fibers. Their target distance is 0 because they're always going to pull no matter what. And maximum distance, which is going to be long. We don't want them breaking spontaneously. And the strength of the construction. Okay. So it'll take a minute or two for people to type that. I may be able to put that in the chat, but let me see if I can actually give you that code. The answer is no, because it was pasted in as an image. So it's not text. Otherwise, I could cut and paste it. But give me a thumbs up when people have that typed. Alex, if you got all of that typed, maybe you could cut and paste the global and the definitions into the chat so people don't have to retype it.
Yeah, I'll put it there. Let's just hope I spelled everything the same. I think I did.
Well, we'll find out, because if you didn't, you'll get a syntax error when you run it. Try running it. It shouldn't change anything.
It runs for me.
So now, in the step function, what are we going to do? That cell has to create new links, pull, and then delete links. And we don't want it to doing that every time step. But what we're going to do is, what the cell's going to do is it's going to throw out its grappling hooks. It's going to pull for a while. When it pulls, it's going to move. So its neighbors are going to change. And so then we're going to have it release the grappling hooks, throw out again, and pull again. So this is going to be a ratchet. It's going to be a molecular ratchet. Now, in reality, the grappling hooks don't get created and destroyed all at once, the way we're going to do it here. The individual filopodia are extended and contracted and broken, extended, contracted, and broken independently. And so if we wanted to build a more realistic model, each link would be independently created and destroyed. But that's a little bit more work than we have time to do in class today. So what we're going to do here is, within the step function, we're going to wait some amount of time, say, probably, Emma, don't wait 5,000 Monte Carlo steps on your computer. Maybe wait 100 Monte Carlo steps on your computer, okay? And then every 50 Monte Carlo steps, we're going to create a function that's going to delete the links and a function that's going to create new links. And at the moment, those functions are just functions. We haven't given them what's going to go inside of them. And so that won't work. In other words, until we've defined those functions, it's not going to work. But why don't people put that in position for the moment as placeholders? Normally, every time you add something, I would say to you, run it and see if it works. But until we've defined delete links and create links, it's not going to work. This is a case where good coding practice maybe makes the exercise a little bit more cumbersome. Because if we typed all of the code in line, it would run at each step. Here, we're creating placeholders for the create links and destroy links functions. And so it's not going to work until we've defined those. Okay. Let me know when you're ready, and I'll move on from that. The delete links function probably you could figure out for yourself without any prompting. The create links one, you probably could figure out for yourself too, but it's a little more work because you have to measure the angles. And so that's a little bit more complicated. Emma, is that okay for you? Okay, so now for the delete links function, we'll go in and we'll create a function definition. You have to decide where to put the function definition. It can be pretty much anywhere inside the class. And so since we created self.delete links, we have to create our function def delete links. And since it's Python, the argument of the definition is always self. And we're going to go over the cells of type crawler. So you could use the iterator, the visit all cells of a given type. We're going to visit all cells of type crawler. And then if you go down that long list of functions that are given in the focal point plasticity menu, we want to remove all cell FPP links for a given cell. And so if you use the twidit menus, you could paste in the for cell and cell list by type, change the type to be crawler, and then paste in self.remove all cell FPP links. You have to make its argument cell. And so this will go through and delete all of the links that that cell has. Okay. So we've left the hardest part for last here. The hardest part is going to be creating the links. And so we're going to create a function, def create links, self. And what are we going to do with that function? Well, the first thing we have to do is find all the crawler cells. Then we have to make links. And those links have to be based on our neighbors and the position of those neighbors. And this is a little bit, this is now a little bit of trigonometry that we have to do. We're going to take our current cell and its neighbor, and we're going to ask the question, what is the angle? And here we're going to assume that the cell is trying to crawl in the x direction. In reality, the cell should have a polarization vector, and we should be measuring relative to the polarization vector. But again, for the purposes of an in-class exercise, we're going to simplify by assuming the cell's trying to move always in the x direction. And so what we're going to do is we're going to get the vector between our current cell and its neighbor. There's a function that does that for us. We're going to get the angle between our cell and the neighbor. That's returned in radians. We're going to turn it into degrees. And we're going to ask the question, is that angle less than the maximum allowed angle that we defined at the beginning? We said 50 degrees. If it is, we're going to use the create link function to create a new link between our cell and the neighbor with the arguments lambda, the strength of the link, target distance, which we set to 0, and the maximum distance, which we set to 1,000. So basically, if there's a neighboring cell within the range, then we create a link. We're not going to worry about min and max. And in this case, Julia, in his example, is using math, import star, A10(2) of y comma x. You have NumPy loaded. And so there, I'm trying to remember, you have to look up, somebody, maybe Hayden, can look up the NumPy equivalent. It's probably np.a10(2) would be my guess. So you don't have to explicitly from math import star. If somebody sees that up first. It's a minor annoyance to both C and Python that the math functions are not built-ins, that they have to be added. I'll give people a minute to. Again, this looks like a lot of typing if you have to type all of it, but a lot of it is pre-implemented in the pull-down menus, so you don't have to do all. As I thought, it's np.a10(2). You don't have to load math. It's included in NumPy. This slide was probably written before Compusol by default loaded NumPy. So if you do this, the simulation should work. You should actually see a crawling cell. Emma, are you nodding because it works, or is there a problem? You want to show us your result? That's great. You'll notice as the cell moves, the angles, that basically the angle opens up. Sometimes it only gets one link, sometimes it gets two. But it's not that pretty, but you see the cell is crawling to the right. And you'll see the cell fluctuates, the shape of the cell fluctuates quite a bit. And that's actually not that far from what we were seeing when we were seeing the visualization of those individual cells crawling through the tissue. Now we could play with a variety of things. We could make the strength of the link different. I think that if you have written the lambdas in the Python, that it's not going, that I don't think that steering panel will work. I don't think that will overwrite the values. So unfortunately, I think once you've switched to using the Python specification, that steering panel doesn't work anymore. Now, what we could do is we could have a slider that controlled those numbers. So that's something we could do. Yeah. You'll notice there that because you've got a very large force, the cell is breaking apart. In reality, you do see cells occasionally shed little bits of bleb behind them. But under normal physiological conditions, the cell should split in that way. You notice that the place that the cell is anchored is now outside of the domain of the cell because of the split. So that's telling us that in that case, the force we've applied is a little bit too big for the simulation. Great. What other quick thing you could do is create two cells, create two crawling cells. Let them both crawl together. And you'll see if they get too close to each other, they'll interfere with each other in potentially interesting ways. If they're far apart, they'll crawl independently of each other. Okay. Alex, did that work for you too?
It does not like why. It's the present error.
Sorry, I didn't catch that.
His name Y is not defined.
Okay, you want to take a look?
Y is not.
Okay. Can you zoom in a little bit? Because let me see. I could probably zoom on my screen. X, Y, Z. That's interesting. So it's failing in line 40. So why don't we see what is returned by line 39? Why don't we try printing X, Y, Z? Presumably this should fail too if Y doesn't exist. So is it failing in the print statement now? No, it's still failing in the ATAN. So did it print? Can we see, did it print in line 40? Yeah, let's try that.
Oh, it's at the top. That's why.
Oh, okay. All right. Okay, but now you have to undo the comments, I guess.
It's pretty.
Yeah, no, it was just you had the A10 in the wrong place. Let's try again. Let's look at your code. Should be okay now. Does it look like it's creating any cells, any links? Okay. Right. Let's go back to your code. No attribute append 2. Huh. What is that one is baffling to me. Emma, what did you use?
Sorry, I went to the bathroom. What did I use where?
So Alex's code is failing here in line 41 because it's saying NumPy has no attribute A10
I used math.a10
Okay, so let's try math.A10
NumPy does have a10 though. I'm not sure what's going on.
Yeah, I checked that. I mean, I looked it up, and NumPy.A10 should be correct. Okay, where? Oh, because we didn't import math. Something very strange is happening because you're doing from math import star.
I also didn't do that because I don't understand it. So I just did import math, and then to reference a particular function, I would use math dot, the name of the function.
Okay, that'll work. That should work too. Yeah.
I don't really know what that star, whether it imports everything so that you shouldn't have to call the module for, I don't know.
No, yeah, it should. Okay. Let's try that. Let's try it. Get rid of the from math import star. You don't want both, probably. Well, this time it worked. Okay. All right. I'm not sure why the numpy.a10 wasn't working. That seems strange to me, but anyway. There you go. All right. One of the eternal mysteries of Python library usage. Okay. So I hope that you got something that crawled and that worked okay. If you didn't, you could find it. It's in the demos. It would be demos, lecture 15, 6.2, and then it would be 6.2.3. It's a demo. All right. So now for our last exercise for the class, I'd like to do something a little bit more fun. This will begin to understand folding of tissues. And the neural tube development example that's here on the left is currently the PhD research, actually, of Job in the Netherlands, who's interested in toxicology. He's trying to model chemical perturbations that lead to spina bifida and other neural tube defects in human beings. And in that case, he has a rather elaborate model of the gene regulatory and signaling networks and their chemical perturbations that he could actually model in some detail chemical developmental toxicities that could lead to birth defects. In our case, we're not going to go to that level of detail, but what I want to talk about is the idea of what's called epithelial invagination and folding, which is you start out with a flat layer of cells in a sheet. A group of those cells begin to contract, and they contract on their apical surfaces, so they pull in. And if they contract enough, you could get the formation of a tube like this. And this is a very common process in development. It happens again and again and again. Not only the formation of the neural tube, which will eventually lead to the spinal cord, but in a whole set of different situations. It also happens in fruit flies, in worms, but we're going to try to understand the basics. And again, if you have trouble finding this, it's in module 6_4_1. So what's going to happen here we have an initial set of cells that are uniform. Something is going to change in a subgroup of those cells. And at the apical surface of those cells, there are going to be contractile forces that are going to make the surface area of the cells less. And that's going to make the cells essentially wedge-shaped. If the cells are wedge-shaped, we know that that is how you build an arch. And so you're going to get a bending of the tissue. Now, exactly how it bends depends a little bit on how much tension the whole tissue is under, and also what's on the bottom. In other words, if there's a rigid surface underneath, it won't let the cells move down, then you won't get that kind of invagination. There are other kinds of invaginatory processes that can happen. You could have cells disappear by dying, for example, being extruded. And you could also have changes in the microarchitecture of the cells beyond which are in the microtubules rather than the actin filaments. But this arch formation here on the left is the classic invaginatory process. So what we're going to do is we're going to create a basic epithelial sheet with compartmental cells, and then we're going to create links in that apical surface that are going to pull the cells together. And so the goal of this exercise, and I hope we're able to get through it in the last 40 minutes of our class together, is going to be, and again, if you can't do it, you can pull the code from the online repository. We're going to create a compartmental cell layer like this. We're going to have every cell composed of an apical compartment, a lateral compartment, and a basal compartment. And then we are going to cross-link those apical compartments and make them invaginate. So we're going to create a model template. We're going to adjust the cell areas. We're going to group the cells because we have to make compartmental cells here and adjust their contact energies. We'll do that first without creating the links, okay? So the first thing that you want to do is you're going to create a simulation. It's going to be 350 by 100 by 1, periodic in the x-axis, fixed boundary conditions in the y-axis. We're going to have 4 cell types: apical, lateral, basal, and ECM. We're going to enable focal point plasticity. Then we are going to edit the uniform initializer to make layers. Then we're going to modify the focal point plasticity so that we only have links between apical surfaces in particular cells. And we're then going to have to adjust the contact energies of the cells so the cells don't make a mess. And then we're going to put in the links by hand because we don't want all the cells to be linked. If every cell across the top is linked, we're going to get a uniform contraction. We're not going to get invagination. All right. So please start Twit, start Wizard, give your simulation a name, and we'll make the simulation 350 by 100, periodic in X, rectangular slab initial conditions. And then we're going to create 4 cell types: apical, lateral, basal, and ECM. Somebody give me a thumbs up when you're ready for that. Okay. And if you're stuck, feel free to use need more time or put in the chat, need help, as you like. Okay. As usual in this situation, we're not going to use chemical fields, so we skip the chemical fields. And we select contact. Now we're going to have compartmental cells, so we need that contact internal turned on. Focal point plasticity, because we're going to have links. Volume flex, we're not going to be having the cells grow or divide during this simulation. Although the simulation is much more interesting when cells grow and divide. One of the places that the clone parent from child to child doesn't work is links. So when you have cells divide, you have to redo the links by hand. It's too fundamentally, there's no default way that links should inherit. And so when you have cells divide, you have to redo the links. We'll have center of mass and cell neighbors. So why don't people finish that, run it, and tell me what you get. It's not going to be very pretty. Certainly not going to look like what we want. You get something that looked like that? Emma?
I made the layers, so no.
Oh, you've already made the layers. Okay, so you're ahead of us. Okay. Alex?
Yes.
Okay, great. So as Emma said already, the next thing we have to do is take the uniform initializer and turn it into layers. There are varieties of ways you could do this, but the simplest one is to just by hand put in the layers. So we could set up our ECM layer, box mid of 0, that's this bottom left-hand corner, box max 350, the upper right. Make the layer 35, and we'll make these ECM cells big, 35 by 35. And when we do that, we just get the ECM cells, nothing else. And now we're going to put a layer of basal cell type on top of that. So between 35 and 42. And here I'm making the basal cell layer 7 by 7. And now I'm going to make another layer of square cells that are those lateral cells, and then the apical cells. If you picked a different size, it's not a big deal. If you made the basement membrane, the ECM, 5 by 5 or 7 by 7, it's not going to make a huge difference. But we do want to make sure that the lateral, basal, and apical cells are the same width, because we want them to be stacked on top of each other, because we're going to group them in clusters vertically to make single cells. Okay. Any questions about that? I'll give people a minute to make sure that that works. Is that how you did it, Emma, or did you try to do it a different way?
I just tried to divide the space evenly between the 5 types, but then I realized that doesn't make sense, so I did it this way.
I mean, you could certainly make the ECM layer thinner or have it narrower if there's no. The only thing is, since we can only draw squares using uniform initializer, we're sort of stuck. If we want to make it thick, we'd have to make multiple layers of ECM, which is okay too. The ECM is really just passive in this simulation. It's just we want some kind of support, but it doesn't do much. Alex, how about you? Is that all right?
Yes.
Okay. Then when we go into the volume term in our XML, if we run this by itself, we'll see that the default volumes are 50. And so the ECM will go shrink. And so in the volume plugin, we have to make the target volume. We had it as we made it here. It's actually 35. So 35 squared. I believe him if he says 35 squared is 1225. Maybe I shouldn't believe him, but you could check. Okay. And then you should get something that looks like this. If you don't see the FPP links, you have to remember to turn them on in the visualization. So do people get something that looks a little bit like this? Depending on your screen resolution, you may or may not be able to see the little white lines that represent the ECM links. I mean, the FPP links. So we're getting closer. We're not very close, but we're getting closer. If that's working for you, give me a thumbs up. If you need help, let me know. Is that okay, Emma?
Having problems with my ECM cells. They're
Oh.
way too big.
They're
Also-
too big. The 1225 is too big, is wrong.
Well, I also use different simulation dimensions, so because I'm trying to make it faster.
So you made a smaller. If you cut the simulation off, you need to make the vertical dimension, you really need it to be about 100. The X dimension, if you made it, say, 0 to 150, that would probably work okay. We picked the dimension so that there'd be an even number of cells. So we made it 350 because we knew we were going to make these cells 35 across. So if you made them 20, you could, if you wanted to make it 200, you could make the ECM cells 20 by 20, and then a target volume of 400 would work. This is actually a pretty fast simulation. So we're not using chemical fields, so it should run pretty fast. Were you able to get that, or are you still working on it?
One thing I've actually never been able to understand is what the different initializer parameters are. What actually is width? It's not one side, is it, of the cell?
So the cells are always square. So if you say width, if you say width of 20, it'll draw 20 by 20 square. If you say gap, it'll put a space between the cells of that width. It would be nice if you could draw fancier things like rectangles or actually the newest version of CompuCell has you could draw circles with where it draws little wedge-shaped cells. Peter Fife wrote that for us. But historically, when people wanted fancy layouts, they did it in Python in the start function. And so since there were so many possible layouts that people might want, we just had these two very basic ones. Another one that you really might want would be a hexagonal grid of cells, where instead of square cells, the cells were more or less hexagons. Or you might want a random initial condition of cells. And that's also useful. And so I agree with you that we could provide better initializers than these. One thing that you could do, which we haven't really talked about, we haven't done much with file handling, but it's not hard to learn, is if you run a simulation to say start with the squares and relax them to be hexagonal, you could save that file as an initial and then read it in as an initial configuration. So you could output the simulation conditions and then pull them back in. Then you don't have to draw them each time. Okay. Is that working now, or do you need? Okay, great. So the next thing we want to do is we want to apply this contractile force along the top. And to do that, we're going to first have to build cluster cells where we take the apical, basal, and lateral surfaces, materials, and put them together. And then we're going to have to create links between them. And so a reminder on focal point plasticity, we have that contractile strength, lambda. We have the target length of the links, which is the desired distance between the two cells. We have the breaking length and the number of links. So in this case, we don't want to have them created automatically. And so in our code, in our XML code, we'll turn local on, and then we're going to delete all of that other code that was generated automatically. Okay. So we just want plugin name focal point plasticity, local, and then end plugin. That's it. We're going to do everything else in Python. So now when you run it, you should see no links. You shouldn't see the links that we had before. You should see that. Well, here, sorry, I said the wrong thing. I got ahead of myself. You're going to delete all but the apical links. I apologize. I said to delete all of them, and I'm wrong. Delete medium apical, medium lateral. Again, Wizard tries to guess what you want, doesn't always guess right. Leave the apical ones, and then run that. And so now you should see something that looks like this, where you have horizontal links between some of the red cells. Does that work? Yes, no, maybe. Yeah, okay. Okay. And now we're going to turn those extra links off, and we're going to do that by saying maximum number of junctions is 0. So what are we doing with this focal point plasticity plugin? We could do everything by hand, but we're going to do things partly by hand and partly not. We're going to create a placeholder that says we can link apical to apical, but we're going to set target distance, and max number of junctions to 0. So it means there's a placeholder for creating the links between the apical compartments, but we're not going to specify the parameters. And it's not going to define, and we're not going to create any. If you set something differently, it's okay because we'll overwrite it later. Now, we have to change the external contact energies. We want apical cells internally to stick to lateral and basal, because we want them to stay in the middle. Sorry. Actually, is that right? Let me see. Apical to lateral should be low, I would think apical to basal should be high because we don't want them touching each other. Lateral to basal should be low because we want it to touch. So I might disagree with the setting here that Julio came up with. I might have set that one to be high. And here are the suggested energies that he's going to use. If you look at contact internal, you'll have a big table. We'll set apical to 0, apical lateral to 2, apical basal to 2, apical ECM to 0, lateral to 0, lateral basal to 2, lateral ECM to 0, basal to 0, basal ECM to 0, ECM to ECM 0. ECM is not going to be part of the cluster, so in fact the ECM lines could be deleted. They're not doing anything. But basically we want, and the self types, apical will never occur because you're only going to have one apical compartment per cell. So in fact, a lot of these things don't ever happen. The only things you have are apical lateral, apical basal, lateral basal. Those three are the only energies that actually matter. And here they're all being set to 2. Okay. Now for external energies, it's a little more complicated. Medium, which is on top, has a low energy with the apical surface. That's how epithelia work. The apical surface has a low contact energy with medium. Medium has a high contact energy with lateral surfaces and basal surfaces. Doesn't like to touch anything except the apical surface. The apical surface sticks to other apical surfaces, but is repelled by lateral and basal surfaces. Medium does not touch well with ECM, and it also repels, and apical surfaces also repel ECM. ECM wants to be at the top. It should touch medium, and it should touch lateral and other apical, so its own lateral. It should touch laterally the other apical surfaces. Lateral sticks to the other lateral cells. It sticks on the side through junctions and adhesion. It doesn't stick well to basal on the other cells, and it doesn't stick very well to ECM either. Basal sticks to its neighboring basal compartments and to ECM. And so this time we have to define a bunch of energies. Again, the medium adheres to the apical surface, repels everything else. Apical adheres to other apical and repels everything else. Lateral adheres to other lateral, repels everything else. Basal adheres to basal and to ECM. And so here are the energies we're going to use. And you're going to need to go through and type those in. Medium doesn't do anything, so it doesn't matter. Medium apical 5, medium lateral 10, medium basal 20, medium ECM 20, apical 5, apical lateral 10, apical basal 10, apical ECM 20, lateral 5, lateral basal 10, lateral ECM 10, basal 5, basal ECM 5, ECM 0. That'll make the ECM mix. You could make that 5 too. And run that and see what you get. Let me know if that works. Okay, Emma, you're nodding your head. Okay. The last thing we're going to have to do, the two things still, is create the compartments. And the compartments are going to be created in the start function. And so what we're going to do here is we're going to loop over the apical cells. And we're going to check if they're in line vertically. And if they are, we're going to put them into the same compartment. And I might actually have done this a little bit differently. But this is how Julia does it. So we're going to do it. So what we're going to do is we're going to loop over all the cells of type apical. And then we're going to go through all the cells of type lateral. And we're going to ask if the cell of type lateral is the same position as the cell of type apical. We're going to put them in a cluster together. And my cats decided to knock everything off my desk. If you hear loud noises, and that's this is a little bit risky because if the cells weren't exactly aligned, it's going to fail, right? We're depending on the fact that when we'd use that uniform initializer, it was putting the cells exactly in line. Another way to do it would be to check whether look for the neighbors of the cell. And that might be more reliable, but let's use Julia's code for the moment. So I'll let people type that. Can people see that all right, or do I need to zoom on it? I'll let you spend a second typing those loops. Again, you could use the twin iterator function so that you don't have to type things quite so much by hand. But we're using that self-reassigned cluster, which is in the cluster menu for twin. I'll give people a minute to implement that, and I'm going to clean up my cat's mess for a second while you're doing that. Now, when you run this, it's not going to look different. The simulation shouldn't look different when you run it. Did it work? Did that one work? Did people get that to work? Alex, Emma, is that all right?
I'm changing dimensions.
Okay. I mean, there are a variety of ways you could do this. As I say, iterating over all the apical cells makes sense. But if I were going to write this myself, for each apical cell, I'd look for its neighbors. I'd look for the lateral cell beneath it. And then I'd iterate over those lateral cells, look for the ones next to them as well. But this certainly will work. The other thing you could do is instead of saying if cell.xcob is equal to cell2.xcob, you could say if the xcob is within some tolerance, ±2, for example. That would allow you to build a little bit more flexibility into the code. Now, something that's in this slide, which was on the slides that I showed, but I didn't really talk about, is I gave the impression perhaps that cells don't have a cluster ID until you assign it. And by default, cells have a cluster ID, which is their cell ID. And so you might expect that this would fail because cell.cluster ID wouldn't exist. But in fact, the apical cell automatically has a cluster ID, which is by default the same as its cell ID. And so you don't have to instantiate the cluster ID. Although you could do it that way. You could create a counter for the cluster ID and then create cluster 1, cluster 2, cluster 3, and so on. In this case, the cluster IDs will be whatever the apical cell IDs happen to be, so they won't be sequential. So give me a thumbs up. Okay, Emma, it looks like it works. Okay. So now the next thing that we have to do is create those FPP links between our apical cells, our apical now compartments of those cells. And so we're going to iterate over the cells of type apical. And now we're going to go for neighbor in common surface area, find the neighbors of that apical cell. If the neighbor happens to be also of type apical, that's the left and the right, then we're going to create the link. Okay. So then we're going to go in and we'll create link, self.new FPP link. That comes from our menu. Between the cell and the neighbor, we'll make the strength of the length 7, the target length 7, and the breaking length 200. And when you run that, you should see a white line, or black, depending on the color that you've used, running all the way across your simulation. Okay. And so you now have an epithelial sheet of cells. So the last thing that you want to do is you want to make it bend. And the way you're going to make it bend is you're going to relax the cells. And so you're only going to put those links in for certain cells. So now, at time 500, you'll go and you'll say if the X position of the cell, an apical cell, is between 125 and 225 in the middle, and if you made it shorter, you'll have to change the numbers. Now, you're going to change the properties of those links. And you're going to change the properties of those links by changing the lambda distance. Pedro says we're out of time. So give me 2 minutes to walk through this, and I'm not going to make everyone do it by hand. You're going to set the lambda distance to be different. You're going to make it here to be larger. You're going to set the target distance to be, sorry, the lambda distance makes the cell, the force very large. Apologize. You're going to set the target length to be short, to 3, and then you'll get that. So let's try that. Let's take 2 more minutes and make that work. And you'll have the invagination. And that's a good place to end, I think. Okay, so here's the code. You're going to go through the cells. If they're in that middle region, you're going to get the link. It's connecting the cells. You're going to make the lambda link large so that they're strong, and you're going to make them have a tension and pull. Okay. Emma and Pedro had to go. Alex, does that work for you?
I'm almost done typing it. Just one more line. That's I think I typed something wrong.
Okay, do you want to take a look? Why, take a look here, see if we get to work it. If not, we'll look at it together.
Oh, I see what I messed up.
But this in some ways is very basic, but it's a very powerful framework to use because we could now start with this. We could actually make cells that will make a real circular invagination. We could make it pinch off. It's not hard to do that. We could have the links controlled by a network of signals so that the bending happens in response to external chemical signals or internal cell differentiation. And basically with this level of detail, you could build very complicated mechanical simulations like that one of the formation of the neural tube. Now, this particular approach works better in 2D than 3D. As Alex, as you know well, when you do it in 3D, the problem is you have to link to a lot more neighbors. And so the connections become a little more complicated to set up. But in 2D, it's pretty easy to make. Did it work for you, Alex? Still an issue?
Yeah, it works.
You want to show us?
See if it works.
Okay, that's not the prettiest thing in the world. My guess is that what's happening here is that the energies for lateral compartments are not defined quite right. But the basic structure is working pretty well. If I do it, this is what I wind up with. And so I'll leave it to people to see how to get to that stage. The code is there if you want to try to load it. And I think Alex, it's almost certainly an energy. One of the energies in your probably the contact internal isn't quite what we expected. Okay. So that's where we're going to leave things. You could play with changing lambda distance, which is the strength of the links. You could change the area over which things happen, the number of times you have this, because sometimes you'll get clustering in multiple locations at once. But at least today I wanted to show you a little bit of the mechanical possibilities. We played for the past few weeks, we played with molecular control. I wanted to show you some of the kinds of mechanical control that we can implement in CompuCell. Well, I appreciate everybody showing their projects as they were today. Please, two teams meet together, go over each other's projects, critique them, submit those to me. And then be ready next week at 5 o'clock to do your final presentations and upload everything. There are slots for the preliminary uploads that have been put up on Canvas. I'll put the rest of them as well. If people have additional homeworks from the later groups that they want to submit by Friday, I will grade everything over the weekend for people, but that'll be it. After Friday, there'll be no more homework submissions at all. Please do focus on the projects going forward. So I want to thank everybody for participating in the class. I look forward to seeing you next week for final presentations. And that's it for today. Thanks so much.